Well, thank you very much. It's a great pleasure to be here. Yeah, let's move to a group who has a little bit more wisdom, I would say, so to all the adults. This is really about cognition in old age and how to differentiate cognitive problems in old age from incipient Alzheimer's disease. Kind of an important topic, obviously. This is a collaboration between UCL and the German Center for Neurodegenerative Diseases, a Brexit-defying fight against Alzheimer's. So what I would like to talk about is basically what happens as we age to our memory, and how can we tell apart whether what we experience maybe as memory problems is just related to age or has anything to do with Alzheimer's disease, the most frequent form of dementia, and where aging really is the biggest risk factor. Now, first of all, obviously, just aside from Alzheimer's disease, it's clear that our cognitive abilities in old age are influenced by a lot of lifestyle factors. So nutrition is very important, mental stimulation is very important, physical exercise, social inclusion, and obviously there are a lot of health factors that have an influence. So our brain needs a lot of glucose, so insulin resistance is a risk factor. Other metabolic risks such as obesity and hypertension. It's important that the vascular system of the brain works well, and there are also some genetic regulators that have an influence. And these are just a few of the factors that can basically affect how well your memory and how well other cognitive abilities work. But aside from these factors, it turns out more recently that also when we just look at individuals who seem cognitively normal and don't have dementia, we need to take into account the changes that we would normally ascribe to Alzheimer's disease. Unfortunately, this is not very well visible, but what you should see here are cross-sections of a brain. So in Alzheimer's disease, we have basically two culprits. These are toxic proteins that accumulate in the brain. One is called tau, and the other is called amyloid. And we know a lot about the accumulation of these toxic proteins in the brain, particularly by the work of Heiko Braak. This was a really dedicated pathologist. He's now retired. He worked in Germany. And he just looked at brains of individuals who passed away for all sorts of reasons, not just because of dementia, but it could be a heart attack, it could be a car accident, or whatever. And he looked at thousands of these brains, and he mapped out where in the brain he would see these toxic proteins accumulate or increase. And so you really can't see this, but this would be half of a brain, and he would see that in some individuals, he would see tau just in a very tiny part of the brain. In other individuals, there would be more brain areas affected, in particular this one here, and so on. So there would be a gradual difference between individuals in how much tau protein would be in their brains. When he looked at amyloid, he found a very different picture. He found that amyloid was very diffusely distributed, but this particular region here, which is actually called the hippocampus and is very important for memory, is actually spared from amyloid initially. Now, what's interesting is when you look at the frequency of these accumulations in the population. So this is a somewhat complicated slide, but the graph. But what you see here is age, and this is the percentage of people who show these types of accumulations. And this particular stage here, where you have basically tau accumulating mostly in the memory regions, when you look at the 60-year-old individuals, you can see that almost 80% of individuals already have tau in their memory areas. When you look at the 60-year-olds again and now look at amyloid, you can see that already 30% of older adults who basically already have amyloid in their brains. Now, when we just look at the incidence of or prevalence of dementia at the age of 60, it's around 5% or so. So already from these graphs, you can tell that there must be a lot of older individuals around 60 who have no cognitive problems or just minor cognitive problems, but carry changes related to Alzheimer's disease. And this is something that's now basically causing a lot of headaches to those who work in the field of dementia, because it raises the question, what is actually aging, and where is the transition between aging and something that has to do with Alzheimer's disease? So that's really a major challenge that now the researchers are facing. Now let's look at the changes related to Alzheimer's in a bit more detail. So this is a MR scan of a brain. Let's magnify this and take a piece of cortex. Now, if we basically then magnified this, obviously this area would contain neurons, and this amyloid proteins would basically aggregate as plaques. We call them amyloid beta plaques, or as strings that are called oligomers, and the tau proteins would accumulate in the neurons. How can we measure this? We don't want to always look at post-mortem brains. We want to actually be able to easily measure these changes. And one way to do this is just by looking at the spinal tap, because these proteins are also appearing in the cerebrospinal fluid that basically here is that circulates around the brain, and you can measure this in a spinal tap. Neurologists like to do this kind of thing. It actually doesn't hurt so much. It looks much more painful here. But you can also do a positron emission tomography. So we have meanwhile been able to actually create radioactive substances that bind to these toxic species. And then you can visualize where in the brain you can find that type of plaque, or where in the brain you can find these types of tau proteins. And with that type of information, we can now we are just now starting to understand how cognition changes, as there is basically a gradual accumulation of these changes in the brain. And it's clear that at some point, as you start accumulating amyloid and tau, cognition starts declining. The decline at some point then is called mild cognitive impairment. This is when only memory is affected. And at some point, it's so severe that we speak of dementia. And then somewhere before the mild cognitive impairment stage, there's a stage of subjective memory complaints. Now, the challenge is that this stage, where you actually already have some amyloid and tau in the brain, but don't actually have anything related to dementia, anything that would qualify as dementia, is a very long stage. It could last 20 years or more. And here, basically, when we speak of cognitively normal older adults, we have to face the situation where we know that there is already that pathology in the brain. Now, in order to be able to work with this, there is now experts from all over the world got together and basically came up with a very complicated way to actually try to classify this. And it's got to be complicated because it is a very complicated matter. You have tau, you have amyloid, there can be different combinations of these things and so on. And at the end, what came, the classification that basically comes out from this is that you need to have both amyloid and tau in the brain in order to, in order for this to be called some preclinical, very early stage of Alzheimer's disease. If you have just amyloid, so here's a patient who has just amyloid, no tau, and a completely normal brain scan, then that's not yet Alzheimer's disease. Or if you have just tau and no amyloid, that's also not Alzheimer's disease. So in our research, we are particularly interested with the consequences that tau has on memory function. So let's look at this again. According to all what we know, when tau just affects these memory areas in the earliest stages, really these are the stages where we would say these are age-related memory problems. It's only when tau has spread to other brain regions, to a lot of brain regions, it's only then that it is likely that there's also amyloid in the brain, and these are the stages that are related to Alzheimer's. So what happens when you have just a little bit tau in these early memory areas? This is basically what we are interested in, and this is what we believe explains a lot of the variability that we see among older adults in terms of memory problems. So the memory that's mainly affected by when these brain regions where tau accumulates are affected is called episodic memory. It's the memory for personal events, and it's different from semantic memory and from procedural memory, like playing golf in this case. And what's so special about personal events is that these are memories where you have only one opportunity for encoding. You cannot basically repeat this rendezvous every evening. There would be some social problems associated with that. So these are really things that are entirely novel. So this brain system reacts to unique novel events, and it's a very rapid encoder. And so we want to understand how early tau pathology in people who have basically who do not have Alzheimer's disease yet, affects memory. Now, you can obviously look at a lot of different aspects of memory. You can look at memory capacity, you can look at memory precision, you can look at the flexibility of memory. Can you retrieve a certain piece of information when some is missing? And you can look at stability over time. And when we look at this region, the hippocampus, in more detail, we know actually meanwhile from cognitive neuroscience, and a lot of the great research on that is done here at UCL, that all these parameters map onto certain aspects of this complicated circuit and small brain area. But it turns out that what's particularly important in this regard, with regard to tau, is memory precision. And memory precision actually allows us to remember whether we were yesterday, we had this wonderful bargain of the fresh strawberries in this supermarket or that supermarket. Obviously, they look very similar, but the precision system here in the hippocampus allows us to form different memories for these very similar events. Now, when we unfold this structure a little bit, and here is basically a three-dimensional structure, redrawing actually. It turns out, and this is something that we have recently discovered, is that it is actually a bit more complicated than we initially thought. And it has the capacity, it is organized in different processing pathways. So when I look at this place here, obviously there is a room with with the boundaries. There are a lot of objects, and there is a wonderful audience. Now this system breaks this down into just the spatial room aspect and the objects. Somewhere there's also the humans process, but let's not care about that. So the objects and the scene information is actually broken apart into different pathways. And it turns out that tau accumulates particularly in this area that is responsible for the objects. And in order basically to see whether tau accumulation has a specific effect on object memory, we created a task where, that probes memory precision. And it's actually a challenging task. So you see an object like this, and afterwards, when it's repeated, you have to say, oh, this is not exactly what I saw. This is actually a new version. And we do the same with rooms. So here, the boundary of this room is slightly shifted, and you have to basically recognize that. And then we put our older participants into an fMRI scanner, and they basically complete this task. And so we gain some information. Now, the important thing to be clear about here is that we don't actually know how tau affects memory. And so we know, of course, that the memory problem is a consequence of brain dysfunction. But where does the brain dysfunction come from? So many people who work in this field would say, well, it's clear, tau causes loss of neurons, loss of brain substance, and that loss of brain substance causes brain dysfunction? Well, there's another possibility, namely that tau causes directly brain dysfunction because it's slightly toxic. And this is important because, if it's a direct effect, then obviously this means it is reversible. Because if you take tau away now, and you haven't lost any brain substance, then you have a reversible memory deficit. So it's very important to understand whether the effect of tau on memory is a direct effect or is mediated through neurodegeneration. So in order to disentangle this, we take spinal taps from our participants, we measure their brain activity, but we also measure the volume of the brain in order to look at what we call neurodegeneration, substance loss in the brain. This is basically a study that we do at the German Center for Neurodegenerative Diseases, which spans a lot of different German sites, 10 universities, so it's a huge study. And people actually do this type of fMRI task at these different sites. They also do a very simple, more complex photographic memorization task. So this basically informs us about the whole episodic memory system, and here we break it down into the scenes and the objects. Now let's have a look first at this memory task for these complex scenes. How does it work in the scanner? So it's actually quite simple. You lie in the scanner, you see photographs like this, and all you have to do is it an indoor or an outdoor picture? It's a very simple task. It's actually also fun to do. These are interesting images. When you come out of the scanner, we actually ask your memory, probe your memory. We show you pictures and ask you, have you seen those before or haven't you? Now it turns out that the amount of tau you have in your spinal fluid correlates with your memory performance in this task. The higher your tau levels, the less accurate your memory is. Okay? Now where does this correlation come from? When we look at the brain activity, we see that the novel images activate a lot of brain regions, and this also includes this area that I mentioned, the hippocampus, and this adjacent region that is specialized for objects. And it turns out that the novelty response, where you look at the tau levels in the spinal fluid, correlates with tau only in the hippocampus. So the only brain region in the entire brain that correlates with the levels of tau is this hippocampal area. Meaning, the more tau you have in your spinal fluid, the less active your hippocampus is to novel information. And this goes along with less accurate memory for the images. And this is not mediated by volume. So we can statistically control for this. So we can see, is the volume of the region actually driving the effect? No, it's not. Meaning that there must be a direct effect of tau on the function. Which means, well, this is important because this is a potentially reversible effect of tau. So it's kind of encouraging. Now we broke this down also into the object and scene task, and what we see is that in the object task you have a correlation with tau in the sense that the higher the tau levels, the more errors you make in the object task. But in the scene task we don't see any relationship to tau, we see a relationship to amyloid, which I don't want to talk, it's too much detail for this talk now. We have also done a PET study where we actually used this radioactive tau ligand where we exactly know where it binds to the brain. And again, the amount of tau accumulation that we can visualize with PET in these regions correlates with the performance in the object task, but not the scene task. So what does this all mean? So these are, our preliminary interpretation is that as we age, we have a problem in object processing, which is related to tau. And then progressively, when the pathology spreads, and probably also when amyloid pathology comes on board, this causes problems also in scene processing. Now, these are obviously very abstract, illustrations. What does it mean, with the more real-life kind of photographic, types of impressions that we are usually faced with? And that's a very interesting aspect, very interesting question. How does this relate to your performance on memorizing, photographs, for instance? Now, it turns out that there's something called memorability. Memorability is a very interesting concept. It's a concept that comes from the observation that if you show people a lot of different images, some images are more likely to remember among everyone, and some images are more likely to be forgotten among everyone. So there's this thing called memorability. We don't yet fully understand where it comes from, but it turns out that people who are actually progressing towards Alzheimer's disease have no problem in memorizing these types of images, but they forget almost entirely this type of image. And what we believe is happening is that here you have a lot of objects scattered around a scene, and it's very hard for the brain to basically process all the objects accurately and place them into their environment. Now, in order to be able to further this type of understanding, we need actually computational approaches. Sorry, you should see some brains here again. And so computational modeling of brain activity and behavior and all these different stimulus features. And this is something that we are doing now together with Peter Seidman. So a lot of these data are now coming to UCL, and he's going to a very bright person at the Wellcome Trust Institute for a Center for Neuroimage, who will now basically model this complex network. Now, this obviously is not an exhaustive list of brain regions and phenomena that we have to understand in order to understand the transition between aging and dementia. Another very interesting area is called the locus coeruleus, a very small region in the midbrain. It's very important because this very tiny structure produces all of the noradrenaline that we have in the brain. And it turns out that just like the areas that accumulate tau that I've shown you, it's also one of the areas that accumulates tau very early on. It's actually among the first areas that accumulates tau. And with a large team of people from Germany, the Institute, the Division of Psychiatry, and ICN, we are now looking into the role of the locus coeruleus also in this transition between aging and Alzheimer's. So to conclude, with modern imaging tools and advances in cognitive neuroscience, we can now look at the transition between aging and Alzheimer's disease much better than we used to. We are moving towards a molecular understanding of Alzheimer's disease, but I think the important message I would like to give today is that we are also moving towards a molecular definition of healthy brain aging. So this is something we haven't really taken on board so well up to now, but this is really a paradigm shift, I think, at least in this type of aging research. I think this will ultimately improve our ability to identify and consult individuals who are at risk of Alzheimer's disease, but it will also improve strategies to improve cognitive abilities in older adults who are not yet at risk of Alzheimer's disease and who don't have Alzheimer's pathology. And with this, I would like to end and look forward to your questions. Thank you.